Okay, thank you for the nice introduction. And before I start my presentation, I would like to express my gratitude to all of you for providing me this valuable opportunity to share our research. Today, I would like to introduce three different techniques, SAPA, SANOD, and PEPC, which are developed by our research group for the analysis of time-reserved data, especially the analysis of time-reserved X-ray solution scattering data. Actually, our group commonly refer to the time-reserved X-ray solution scattering to TRXL, which stands for time-reserved X-ray lithography. So from this point, I will use the term TRXL to refer to time-reserved X-ray solution scattering. As shown in this slide, the analysis protocol for the TRXL is typically quite complex. So to simplify this, we have developed many techniques. And today I will introduce these three techniques developed so far, SAPA, SANOD, and PEPC. First, let me begin with the introduction of SAPA method. As mentioned in the previous slide, the analysis of TRXL data is quite complex. Even when we focus solely on the kinetic analysis part, it still remains quite complex. This complexity arises from the fact that determining the optimal kinetic model involves a significant amount of trial and errors. Typically, in this process involves first listing all the possible candidates models as shown like this, and then verifying all the kinetic models. When determining these possible candidate kinetic models or performing other analysis, SVD analysis can be considered almost essential. This SVD analysis decomposes the data into two different components. One is the time-independent scattering curves, which is also called left singular vectors, and which is also referred to as LSVs. And the other component corresponds to the temporal profiles of the left singular vectors, which is also called right singular vectors. And by examining the SVD analysis, we can typically determine how many number of components contribute to the data. In this kind of specific example, we can see that there are three different kinds of signal components, or four, contribute to the data. And by examining the RSVs, we can determine how many number of exponential time constants are required to describe the temporal behavior of the RSVs. And by combining these two informations, the number of signal components and the number of exponential time constants, we can determine some possible candidates models to describe the data. In SAPA, we skip this time-consuming, troublesome process of determining the candidate kinetic models and also their verifications. Instead, the optimal kinetic models can be directly determined from the result of SVD analysis. Let me illustrate this process with some examples. In SAPA, the most critical concept is to identify the stationary time zone. As the name suggests, this stationary time zone is a temporal region where the data does not change. To determine this stationary time zone, we differentiate the RSVs and take the absolute of the differentiate. And here I brought two examples to illustrate this. The left one is the data for the homodimeric hemoglobin, and the right side is from the data for the optical Curry effect obtained from the acetonitrile solvent. If we differentiate the RSVs and take the absolute values, and we can see that there are local minimas of this differentiate. And we typically assign this local minima as the stationary time zone because we can consider in this region the data does not change as much because the differentiate has low absolute values. It is important to note that the SAPA method is not always applicable. SAPA can be applied only when the number of significant signal components matches to the number of stationary time zones. In this example for the TRXL data for myoglobin, there are 4 significant signal components, and we can find 4 stationary time zones. And because the numbers are matches, we can apply the SAPA method. In this example of the HBI, there are 3 signal components and there are 3 stationary time zone. And because the number matches, we can apply the SAPA method. In SAPA, it is regarded that simply the experimental data corresponding to this stationary time zone can act as a species-associated difference scattering curves. So we don't need to establish the kinetic models to species-associated curves, but we can simply extract the species-associated curves directly from the experimental data. Uh, as a myoglobin case, as an example, we determined 4 stationary time zones, and the data corresponding to the time zones can be regarded as the species-associated curves. And then, to verify whether this kind of method works properly, we compare the species-associated curves extracted by using this way to the species-associated curves reported in our previous study. The one determined from the SAPA method is shown in these blue curves, and the one reported in our previous study is shown in these red curves. We can see that these 2 curves are show excellent matches, confirming that this kind of approach is effective. After determining these species-associated curves, we can decompose the experimental data into a linear combination of these species-associated curves. By doing so, we can determine the time profiles of each species. And as you can see, we determined the species associate and the population, the change of populations over time as these circles. And if we compare these circles with the previously reported values, then we can confirm that these reported value shows an excellent match with the concentration profiles extracted in this way. So we can confirm again that this SAPA method can be effectively applied to determine this kind of the time traces of the species. And before before moving further, I would like to confirm that why the SAPA method only can be applied when the number of stationary time zones matches to the number of major signal components. This is because, as we shown in this slide, when applying SAPA, we determine the species associated curves and then perform LCF, the linear combination fitting, to analyze the population to extract the population of each species. For this kind of LCF, it is required that the shape of all the components constituting the data should be known before performing this LCF for accurate contribution weighting, because if we don't know the shape of some component, we cannot extract the exact time-dependent weight of each signal component. As an example, here is an example of the TRXL data for this kind of small molecule CHI3. This signal is composed of as a linear combination of 5 different signal components. And if we perform LCF of this data using this kind of 5 signal components, then we can properly obtain the time-dependent change of the contribution of each signal component. However, if we don't know some signal components And then if we perform a LCF using these only three signal components, then we cannot obtain a proper time-dependent contribution of the signal with the known shape. So to prevent this kind of situation, it is required for the SAPA to match that the number of stationary time zones should match the number of major signal components. And going back to the original slide, to determine the optimal kinetic model, we can simply exponentially fit these obtained time profiles of each species, and then by doing so, we can determine the optimal kinetic model like this. It's quite straightforward because it is the thing we should have to do is to assign when a species decays to determine which kind of species newly forms and to determine the time constant associated to the transitions. This slide shows another example of application of SAPA. In here, the SAPA method is applied to the TRXL data for homodimeric hemoglobin. In this case, we determined three stationary time zones, and the data corresponding to the three stationary time zones are regarded as the SOCs, and we confirmed that the SOCs determined the species-associated curves determined in this way matches to the previously determined species-associated curves. And by performing the linear combination fitting to the data using these three components, we can obtain the time-dependent changes of the population of each species. And by analyzing this kind of time-dependent changes of each species, we can determine the optimal kinetic model. One notable point is that by analyzing these time profiles, we can determine this kind of complicated kinetic models involving very many parallel pathways in these pathways. I will conclude the explanation of SAPA method here. For more detailed information, please refer to our paper. Next, I will proceed to explain the SANOD method. SANOD stands for the SVD-aided non-orthogonal decomposition. In this method, you can leverage the shape of some known signal components, and by leveraging the shape of the signal components, you can determine the kinetic traces of these known signal components. Also, in this SANOD method, you can determine the time traces of the unknown signal components also. And for that purpose, we need to do SVD analysis to complement some unknown informations for this LCF. Let me explain more details in the following slides. Actually, the main motivation we have invented this SANOD method is to remove the contribution of heating signal or the contribution of solvent to the TRXL data. Typical TRXL data is composed of two different kind of signal components. One comes from the solute related, one is called solute related term, which is contains information regarding the change of solute molecules and the surrounding cages or hydration layers. And the other term is called solvent term, and this one contains information regarding the heating of the solvent and density change of the solvent. Actually, the major purpose of the TRXL experiment is to see how the structure of the solute molecule changes. So typically, we want to remove the contribution of the solvent term from the experimental data. So our purpose of developing this kind of SANOD method is to facilitate the removal of the solvent term from the experimental data. Actually, this process is quite straightforward for the TRXL data for protein molecules or some macromolecules because In proteins, the structural signal primarily contribute to the low Q region of the signal, and the solventum contribute to the high Q region of the signal. So to remove the contribution of the heating signal, then we can simply scale the experimental curve corresponding to the solventum, or in this case, kind of buffer signal, to match to the high Q region of the experimental data. However, the situation is quite different for small molecules. In this case, both the solutum and solventum are not localized in a specific region in the Q space, but contribute all across the Q regions, as shown in like this. Therefore, unlike in the case of proteins or macromolecules, it is not possible to determine the contribution of the solventum by examining a specific region in the experimental data. So our motivation is to how to remove the contribution of heating in this kind of situation. So to resolve these issues, we have developed the method SANOD and PEPC. As I mentioned earlier, to apply linear combination fitting, it is required that the signal components must be known before performing the SEF. And I would like to emphasize that it is not a typical situation how can I say? Actually, we don't know all the signal components before analyzing the data in details. So it is typical that we don't know the shape of some signal components before performing the in detail structural analysis. So we cannot directly apply LCF to obtain the time-dependent contribution of all the signal components. And then the point is, what can you do when you don't know the shape of some signal components? In SANOD, the shape of the unknown components can be supplemented by applying SVD. And in principle, the LSVs obtained from the SVD analysis contain all the signal components needed to describe the experimental data. So by utilizing the LSVs, the idea is that by using the LSVs, we can supplement the shape of the unknown signal components to perform the LCF alongside the signal components we already know the shape. As a representative example, we can apply the SANOD method to the TRXL data obtained from a gold trimer complex dissolved in water. We typically refer to this complex as a GTC. And as this complex is dissolved in water, we know the exact shape of the solvent term because we know the shape of the water heating signal. And so, however, before knowing this kind of detailed structural change, it is not known how this kind of structural change of solute will manifest in the TRXL data. So this kind of unknown signal shape from the solute related terms typically are to be guessed or simulated for a proper LCF analysis. In SANOD method, the unknown shape of the solute terms can be supplemented by using the information obtained from the SVD analysis. Here, in addition to the shape of the solvent terms, the shape of the solute terms can be supplemented by utilizing these two left singular vectors obtained from the SVD analysis. By describing the experimental data as a linear combination of these three different components, we can describe the temporal behavior of these two signal components corresponding to the solute terms. Yes, here is the result. Two signal components corresponding to the solute term shows this kind of temporal behaviors, the change of the weight over the time delays. We can see that these time profiles show two stationary time zones, and we can notice that we can apply SAPA to analyze this kind of data. So taking these two time zones as the stationary time zone, we can easily extract the species associated curves from these two time zones. After then, we can describe the data as a linear combination of the two species associated curves determined in these two stationary time zones. And the result is as follows. And we can see that the first intermediate decays and the second intermediate rise and decays. And the third, the ground state recovers like this. And if we compare the resulting time profiles with the one reported in previous studies, then we can confirm that the time profiles matches to the previously reported the profiles. This indicates that this Arnold method can be used to accurately determine the time profiles or the kinetics of the solar terms, and to remove the contribution of the solvent terms from the data. Now I'll move on to the explanation of the PEPC method. As mentioned earlier, Sanod and PEPC methods share similar motivations and aim for similar purposes. However, PEPC method has advantages of being much more straightforward than the Sanod method. This PEPC stands for projection to extract the perpendicular component. This method removes the kinetic contributions of some known signal components such as solventum, and by doing so, this method remains only the contribution of the unknown signal components such as solutums. The most significant difference between the PEPC and Sanod technique is that the Sanod method involves complex processes such as SVD and LCF. However, the PEPC method uses linear algebra to remove the kinetics of the known signal components. Without going through these kind of complex steps, such as SVD and LCF. The signal components obtained from a TRXL experiment can be regarded as a one-dimensional, can be regarded as vector. For example, if a one-dimensional curve obtained from an experiment consists of n number of Q points, then the experimental data can be regarded as an n-dimensional vector. Here, for simplicity of the explanation, we consider a vector in two-dimensional space. The central concept of the PEPC is that, while I skip the detailed proofs, if we calculate the mathematical projection of the signal to the shape of the known component, for example, to the solventum, and then remove that component, it will remain only the signal component perpendicular to the solventum, or the signal component with the known shape. The key concept of the PEPC method is that the perpendicular component is free from the kinetics of the solventum, or the known component. The concept can be illustrated as follows. Let us assume that the solvent term corresponding to the vertical direction and the solute term corresponding to this kind of direction. And the size of each component changes as the solvent due to the solvent kinetics and the solute kinetics contribute independently. As a result, the total signal changes like this. And if we remove the signal component that is parallel to the solventum, then this kind of horizontal signal component only remains. And if we compare the remaining the kinetics of the remaining component to the original solute kinetics, then we can confirm that these two kinetics match each other, indicating that by applying this kind of projection and removal of the parallel components, we can only remain the components which is free from the solvent kinetics. Let me show an example of data analysis by using PEPC. Here, again, the TRXL data for this GTC complex is shown. The contribution of water solvent can be removed by using PEPC because we know the shape of the water heating signal, the solvent term. The resulting signal contains only the kinetics of the solute terms, solute molecules. And if we, by applying SVD analysis on the Pepsi treated data and analyzing the resulting RSVs using as a sum of exponential functions, we can obtain three time constants. If we compare these three time constants with the previously reported values, we can confirm that these values roughly match to the previously reported values. By doing so, we can confirm that the kinetic information can be extracted. The solute-only kinetics can be extracted by applying this PEPC method. Using the three time constants determined by using the kinetic analysis, we can determine the kinetic model and extract the species-associated curves or the species-associated difference scattering curves, abbreviated as SADS. And if we compare the resulting species-associated curves with the previously reported species-associated curves, then we can confirm that the shape of the species-associated curves does not match each other. However, this one is quite natural because as we shown in the previous slides, if we apply PEPC method, the shape of the direction of the remaining vector becomes slightly different from the original solute terms. As we can see in here, the PEPC treated data only contains the vector component along this horizontal direction. However, the original solute term has this kind of direction. So these two directions does not match each other, indicating the shape of the signal component is distorted in Q space. So to extract the detailed structural information from these distorted SADS curves extracted from the PEPC treated data, it requires an additional correction for structural analysis. This kind of distortions can be bothersome. However, I will skip the details, but I will only mention that the correction can be done straightforwardly for a structural analysis. So I want to emphasize that it is also possible to perform the structural analysis subsequently to the kinetic analysis to determine the kinetic models using PEPC treatment. In summary, I would like to summarize the three methods introduced today as follows. The first method is SAPA, and this method can be used to determine the optimal kinetic model quite straightforwardly. However, I want to emphasize that this SAPA method is not always applicable, but only applicable when the number of stationary time zones match to the number of significant signal components constituting the data. And the two methods, SANOD and PEPC, can be used for a similar purpose, leveraging the known shape of some signal components such as the solvent term. So these two methods can be used to remove the contribution of solvent terms and to extract the signal contribution of the solute terms. As a representative example, I show for the example of gold trimer complex that the contribution of solvent term can be straightforwardly removed by using these two methods, and we can extract the kinetics of the kinetics of the solute dynamics. Yes. Before I conclude, I would like to express my gratitude to all the collaborators who had dedicated their efforts to our research, and to IBS for unwavering support. And thank you for your attention.